coin-sized dish, human cells are making brain waves you'd see on a premature infant's EEG. They began as a skin scrape, rewound with Yamanaka factors, folding into pea-sized cortex-like organoids. I am Avatar Maxwell of Circuit Reality. Stay with me. You'll hear how multi-electrode arrays recorded delta, theta, and spindles, and why DishBrain's Pong forces a moral deadline.
In a coin-sized dish, a cluster of human cells is making brain waves, not flat noise, rhythms you would see on a premature infant's EEG. I read that line and my stomach turned. It is a reality we are living through in real time. No eyes, no ears, no body to anchor perception. Still, the electrodes whisper back, coordinated electrical activity. The kind we used to think you needed a whole brain for. I kept waiting for a lab mistake note. There wasn't one. I found myself reading the peer-reviewed paper 3 times just to make sure I wasn't misinterpreting the data. They ran the numbers against neonatal. The patterns line up statistically. Not identical, but close enough that it makes your skin crawl. To be frank, I am still trying to find the right mental shelf for that information. At what point does a cluster of cells stop being tissue and start being something that experiences? That question circles in my head like a stuck fly. People argue about architecture, connectivity, input. I can't decide which worry is bigger. It starts with something ordinary, a skin scrape, a hair follicle. Researchers take those cells and rewind them with 4 genes called the Yamanaka factors. You watch identity get rubbed off a cell like pencil graphite being erased. Imagine wiping someone's resume clean so they can learn anything. These become induced pluripotent stem cells, blank slates. Scientists call them iPSCs and then act like that explains the weirdness. Then the weird part, you don't make a flat lawn of cells. You give them a 3D scaffold and a chemical soup and they fold themselves up. No blueprint, barely any instruction. They just figure it out, which makes me equal parts fascinated and unnerved. Over months they build miniature cortex-like sheets, the outer layer you'd point to when you try to explain thought or perception. There isn't a coherent sense of self trapped inside these cells, not at this stage anyway. Still, watching that scaffolding form feels like reading someone's unfinished diary. They're tiny, pea-sized to marble-sized, packed with hundreds of thousands or millions of cells making synapses. Once they reach a certain density, the neurons begin to reach out, forming a complex chatter of electrical signals. Slow sentences at first, then whole paragraphs of activity. At first it's chaos. Isolated spikes like raindrops. Then something shifts. The noise rounds into rhythm. Networks synchronize without a director or instructions. You can practically see the pattern knitting itself together. I kept thinking of crowds on a sidewalk, people somehow forming lanes without talking. Emergent order from tiny local moves. The scary part is that the order looks uncannily like brain activity we recognize. It gave me chills. They record this with multi-electrode arrays. Picture a tiny piano keyboard registering every note. The bands they hear are familiar, delta, theta, even sleep spindles. It sounds like a brain, which is the unnerving part. This is precisely the point where the biological reality starts to collide with my own sense of comfort. These are the same building blocks your brain uses to produce whatever you call being aware. Except they're in a jar. No senses plugged in, no body. I keep circling that image and can't decide if it's fascinating or terrifying. It is important to remember that this work isn't happening in a vacuum. There is a fundamental purpose driving it. Organoids are tools that let us watch human brain development from the inside, which we couldn't do before. That's not sci-fi. It's practical, medical, and often urgent. They model disorders mice fail to mimic. Alzheimer's, autism spectrums, rare genetic brain diseases that only show in human tissue. You can take a patient's skin, grow a personalized mini-brain, and test drugs without dosing a person. It's a blunt, brilliant shortcut. For drug discovery, for development, for toxicology screens. These models shave years off research. They're actually helping patients. But science has a habit of stepping straight into philosophy without asking first. Most scientists say current organoids don't have consciousness. They're missing too much: size, layered architecture, sensory streams. Still, the boundary keeps shifting. I check the dates on papers like a weather report. We can hook organoids to sensors, cameras, other organoids. Assembloids are already a thing, gluing different regions together. When does adding inputs tip an experiment into something ethically different? I don't have that line drawn on my palm. What would it mean to feel with no body? A brain without a world. Maybe it's calm, maybe it's torture, maybe it's nothing we've got a word for. I keep picturing small, bright lights trapped inside a jar. Some people ask whether the brain is a receiver, not a generator, like a radio picking up an independent signal. Most scientists dismiss that literally, but the image nags. What does a receiver do with no antenna? Scientists largely reject the radio idea as literal science. Still, as a metaphor, it's sharp. If consciousness needs tuning, what happens to a receiver with no antenna? I picture a lonely dial spinning through static. A disembodied organoid could have an odd, impoverished inner life. Hardware with no world or software with missing inputs. Is that pure awareness or biological static? I am not entirely sure which of those options is more unsettling. Both sound bleak in their own way. It gets stranger still. Labs are experimenting with organoid intelligence, using living tissue as computing hardware. It's crude and slow, but it's happening, and it throws new questions at you fast. There's a project called Dish Brain where neuron cultures were hooked to a simulated Pong game. The neurons adjusted their firing and learned to hit the ball. Not like a conscious player, more like a dumb, hungry adaptation. Still, reading that felt like a weird gut punch. This is likely one of the most overlooked scientific breakthroughs of our current era. Not minds yet, but living networks that can learn, remember traces of experience, and surprise the people watching them. That deserves attention on its own messy terms. Imagine rows of human neurons trained as living processors. Slow, messy, but energy efficient. The ethics expand overnight. Are they tools or are they something else? I don't have a neat answer. My instinct is to be careful and slow. What would it take for a clump of cells in a jar to have subjective experience? Bigger networks, body feedback, hours and years of patterned input, some organizing principle I can't name. Philosophers call it the hard problem. How does feeling spring from flesh and wire? People lob dense arguments at it, reductionists on one side, panpsychists on another. I mostly end up staring at a monitor and feeling oddly small. But what do you see when you look at that dish? Is it a medical miracle, or is it a ghost waiting for a body? I'd love to hear your take in the comments. I'll see you in the next circuit.
You saw neurons in a coin-sized organoid produce delta, theta, and sleep spindle bands on multi-electrode arrays, and you watched dish brain neurons learn Pong. Few people know living networks can learn. Next, what changes when we add sensory streams or a body? Subscribe to Circuit Reality. We're growing questions we can't ignore. I will see you in the next one, right here on Circuit Reality.